Sophie Hill, and I am currently a postdoc in Ethan Goldberg's lab at the Children's Hospital of Philadelphia. But I'm going to talk today about work that I did in my PhD with Miriam Meisler at the University of Michigan, which we just published last month in Frontiers in Neuroscience. So here's just the paper. You can see all the details online. So I do not consider myself an expert on KCNT1. So just as an introduction to those of you who aren't familiar with the sodium channels, the work I'm going to talk about today is relevant to two of them, both of which cause epilepsy. So on the right is a diagram of the sodium channel structure. Unlike KCNT1, it has 4 transmembrane domains, and it functions, we think, as a monomer. So the first gene I'm going to talk about is SCN1A. And as you might know, that's the most common genetic cause of DEE. And in the mouse and human, it's loss of function mutations of SCN1A that cause Dravet syndrome. We think the primary mechanism is through disinhibition, so loss of inhibitory neuron firing. The other gene I'm going to talk about is SCN8A, which kind of works by the opposite mechanism. In humans and in the mouse, gain of function mutations cause epilepsy. And we think that the primary mechanism is by causing hyperexcitability of the excitatory neurons. So the work I'm going to present today was a collaboration with Ionis Pharmaceuticals, who developed a KCNT1 ASO, which has already been published. So Steve Petru has already published on that ASO. It's the same one. But basically, it works by binding the target RNA, which in this case is the KCNT1 gene, and it recruits RNase H1 to come and degrade the transcript. So we're achieving reduction of KCNT1 expression. And we validated this in vivo in our wild-type mice. Here, I'm showing KCNT1 expression in both the brain on the left and the spinal cord on the right. In black are control mice that received a control ASO. And then you can see that increasing doses of the KCNT1 ASO reduce KCNT1 expression by 50 to nearly 100%. So we took this ASO and administered it in mouse models of SCN1A and SCN8A epilepsy. And I'm going to start by showing the survival of the mice that have the SCN8A mutation. So on this graph, I'm showing the age of the mouse in weeks, and then on the y-axis is the percent of mice that are alive. So the solid black line right here are mice that received the control ASO on postnatal day 2, and you can see these mice are very severely affected. They all die around 2 weeks of age. If we gave 15 micrograms of the KCNT1 ASO, we could slightly extend the lifespan. It was about 2 or 3 days, but it was statistically significant. But we saw greater improvements in their survival with 30 or 45 micrograms of the KCNT1 ASO. And you can see at that higher dose, we achieved about 2 or 2.5-fold improvement in their survival. So it's not a long-term rescue, but it is extending the lifespan of these mice, which is exciting. I don't have time to present it here, but we also tried quinidine in these mice, which is a somewhat specific blocker of KCNT1, and we didn't see any effect on their survival. The other mouse model that we tried was SCN1A. So these mice are haploinsufficient for SCN1A. On the bottom here are mice that had no treatment, and you can see about 50% of them die, mostly within this time period between 3 and 4 weeks, which is highlighted in gray. But if we gave 45 micrograms of the KCNT1 ASO, which was our optimal dose in the SCN8A mice, then we didn't see any deaths in this 3 to 4 week period, although we did see 4 deaths in total out of 14 mice, but those all occurred later. So we still think there's a survival improvement in the SCN1A mutant mice and the SCN8A mutant mice. So I guess my question to you, and what I'm interested in hearing everyone's thoughts on, is why this is happening. Because our work is really observational and we don't have any insight from this data into the mechanism. But some possible ideas are recent and some older evidence of coupling of the voltage-gated sodium channels with KCNT1, which is obviously a sodium-activated potassium channel. There's also some recent evidence that loss of KCNT1 generally is just protective against seizures, that the mice die less after being given kainate or electrical stimulation. And then for SCN1A specifically, there's evidence that both NAV1.1 and KCNT1 channels have special roles in inhibitory neurons. So it could be some interaction that's happening specifically in that cell type. Anyway, I'm interested in hearing everyone's thoughts on that. And here's my acknowledgement slide. As I said, this was done in Miriam Meisler's lab, and it was a collaboration with Ionis Pharmaceuticals. Thanks.